Hey guys, today we're going to be talking about microbial mats. So if you've seen my previous geobiology videos, I talked in the most recent ones about microbial weathering, and I talked about how in the next video, which is now going to be a series of videos as well, we're going to talk about microbial zonation. So in this video specifically, we'll be talking about microbial mats and biosedimentary structures. And in the following videos over microbial zonation, we'll go over marine sediments and a whole bunch of topics that fall under that topic in our microbial zonation series of videos coming up here. And you can see some of these topics even have some question marks there, so it's not all certain, but this is the general plan. But for this video specifically, let's just jump right into it. So before I get right into microbial mats, let me just talk a little bit about what microbial zonation is, because you might be like, okay, what does this, broader topic even mean? Well, microbial zonation is actually exactly what it sounds like. It's just the natural zonation of microbial communities. So they kind of layer on their own because of the way that their metabolisms, their metabolic pathways differentiate and kind of are complementary to each other. And we'll talk more about this as we get into how microbial mats develop. But why is studying microbial zonation important in the first place? Well, microorganisms drive elemental cycling on Earth. For example, carbon transfer through the atmosphere, hydrosphere, biosphere, and geosphere is driven by microorganisms and microbial processes, and a lot of this has to do with the diversity of metabolic pathways that exist among microorganisms and how they naturally zone themselves. And that can bring us now to what are microbial mats specifically. And like we talked about in the cell walls and bioabsorption video, microorganisms like to adsorb to solid surfaces. So they like to adhere to surfaces basically. And once adsorbed to a surface, cells multiply and develop colonies. And eventually other species join in until microbial mats are developed. And we go into the more specifics of this and why and how all this happens in a couple slides. But this is the general gist of what microbial mats are, and that is that they're just layered structures that form due to this natural zonation process of microbial species forming these communities of different species layered adjacent to mutually beneficial species, if that makes sense. And this is actually shown here to the right in this figure, a mixed bacterial culture showing the natural layering of bacterial species. And by the end of this video, we'll have talked about each one of these types of bacteria, so don't worry about that right now. But where exactly could we find these microbial mats on Earth today? What types of environments do they like to grow in? Well, unfortunately, these mats are so appetizing to grazing macrofauna that they are restricted on modern Earth to pretty harsh environments like subtidal and intertidal marine environments, hypersaline lakes, as well as anoxic lakes and marine basins that are stratified, and hydrothermal environments, because all of these environments are a bit too harsh to really have a lot of macrofauna that would totally destroy all the microbial mats. However, I want to stress that this is the case for modern Earth. If you watched my Precambrian video or pretty much any of my early Earth history videos, you know that on early Earth, cyanobacterial mats and other types of microbial mats, which eventually became preserved as stromatolites in the rock record, thrived in environments all over Earth. And the reason that they had an easier time to dominate environments on Earth back then is because there were less macrofauna to eat them. But now, as promised, let's talk a little bit about mat development and step by step what kinds of processes cause the initial formation, growth, and full development of microbial mats. First, like I mentioned previously, cells adsorb to a mineral's surface via interactions with surface ligands, and this is bioabsorption. We talked about this in that bioabsorption video, so if you want to go check that out, you're welcome to, but they absorb onto mineral surfaces, and these initial cells begin to multiply and produce EPS to help them better adhere to that mineral surface. And this EPS, which just stands for extracellular polymeric substances, eventually builds a biofilm. And so this community is adhered in this matrix of EPS EPS, which builds into a biofilm, and the film eventually covers the surface, the entire mineral surface, and similar species to those initial colonizers begin to infiltrate the biofilm because they are like, look, we're eating the same things as you, and we want to also reap the benefits of this mineral surface and your biofilm and all of these yummy compounds. And we'll talk in a second about all the reasons that EPS is really beneficial. So hold on to that thought, because I know you're wondering, why do microbes like the EPS? We'll get to that in a second, but basically these similar species join in, and then eventually different species join in that don't eat the same things as those initial colonizers. Instead, they like the metabolites or metabolic byproducts produced by those original colonizers or their sister species. And that allows these different species now to benefit from also infiltrating the biofilm, and they grow these layers because they separate themselves, but they also want to be adjacent to each other because they are mutually benefiting each other with each other's metabolites. And that brings us to the basis for all microbial mats, and that is these mature mats have layers of microbial communities which mutually benefit from each other. Now, when we get to the specific types of mats, like photosynthetic mats versus chemotrophic mats, and all of that, we'll talk about these specific metabolic pathways and the specific compounds that each metabolic pathway is producing that benefits its adjacent microbial community. But in terms of mat development, the final stage is basically the supply of energy and nutrients eventually becomes insufficient to continue supporting the growth of the microbial mat, and growth stops, and eventually the mat becomes broken down by heterotrophs or macrofauna, and that ends the mat's life. Or if it's in a really prime environment for preservation, it could become lithified and preserved in the rock record. And as promised, let's discuss a little bit about the benefits of EPS, or extracellular polymeric substances. These substances have three major benefits for the entire community or matrix of communities. And the first one is that it buffers against environmental extremes. This is why the title of this slide is Mat Microenvironments. This is so cool, and I hope to eventually have a whole video about microenvironments. But basically, these microenvironments are created by the EPS or biofilm because these substances encompass the microbes into this kind of mucousy little world of their own that can buffer against extreme pH, salinity, and toxic metal fluctuations and concentrations in the outside environment, and even desiccation. And so because of this, they're creating their own environment within their EPS that facilitates optimal growth, or at least the survival of the species in pretty extreme conditions. But EPS not only protects the bacteria, but also actively provides diverse functional groups so the biofilm can support a wide variety of microbes in a close proximity, and stabilization of geochemical gradients, facilitating unique niches. So this is kind of for the same purpose. Basically, the stabilization of the geochemical gradients is because of the diffusional properties of EPS that allows different unique niches of bacterial communities to kind of separate from each other in the biofilms enough to not hinder each other's growth, and also to continue to allow the diffusion of nutrients that they need through the EPS into those microbes. And so it's just enough stabilization to keep communities away from each other enough to thrive, but also close enough to mutually benefit from one another, but also not too stable to where they're just immobilizing all the bacteria. They can still move and compounds can still be sequestered, etc. So it's literally like the perfect material that biology has invented to live in these, microbial worlds. And so that's another reason. The third reason here is that it pools and preserves genetic material. This is also true of nutrients and cations that microbes need, but I've talked about that in previous videos. But it not only pools and preserves nutrients, but also genetic material, facilitating gene transfer. And right about now, you might be thinking, oh my gosh, it's like this biofilm or EPS encompassed bacterial communities are one organism. They work so well with each other, they mutually benefit from one another, and not only that, but they also have a canal system acting as a conduit for fluid flow throughout the entire mat so that nutrients can be brought in and waste can be let out into the bulk solution. And this is analogous to a primitive circulatory system, which is only present in multicellular life. So it's almost like these mats are one multicellular organism working together to have the entire group of communities living in harmony. It's so cool. But moving on from general mat development, let's talk a little bit about the different types of microbial mats that exist and how they differ from one another. So the two major types we'll talk about today are photosynthetic mats versus chemotrophic mats. Photosynthetic mats are probably the most often mat type you've heard of, which include things like cyanobacterial mats, and basically they grow in the presence of sunlight and they're typically dominated by photosynthetic organisms like cyanobacteria. And these cyanobacteria can be seen here. These long green filaments are the cyanobacteria. And these will be the types of mats you see in, for example, the thumbnail of this video, where we had that tidal environment with the microbial mats just peeking over the water line there. That is in a sunlit environment. But what about those mats that grow where the sun don't shine? And I mean this quite literally, actually. And that is where sunlight doesn't reach, like maybe hydrothermal vents, for example. Mats can grow, but they don't contain photosynthesizers because obviously there's no photo to synthesize. Therefore, they have to chemosynthesize, and that is why they're called chemotrophic mats. For example, in this picture here, you can see iron oxide encrusted microbial mats at hydrothermal vents. But we'll get back to that. First, let's talk about photosynthetic mats. Cyanobacterial mats are the first type of photosynthetic mat we'll talk about. These mats include layers as shown here on the figure to the right. Basically, it starts with a green layer with filamentous cyanobacteria and sometimes algae and diatoms as well. Then you have a purplish red layer of purple sulfur bacteria and then another green layer of green sulfur bacteria. Now we know that cyanobacteria, algae, and diatoms are photosynthetic organisms, but they use the type of photosynthesis we are all familiar with, and that is oxygenic photosynthesis. They couple sunlight with oxygen to gain energy and release carbon dioxide. However, there are photosynthesizers, meaning they use sunlight, that don't use oxygen in that reaction to gain energy. In fact, the purple sulfur and green sulfur bacteria actually use sulfur and sunlight rather than oxygen and sunlight to gain energy. And because they don't use oxygen, they're called anoxygenic photosynthesizers. This is a whole other category of photosynthesis, and actually they don't love oxygen. And because they don't love oxygen, but they love sulfur, they're kind of deeper down in these mats, where oxygen is less abundant because you had the cyanobacterial layer followed by typically some aerobic heterotrophs, meaning that they respire oxygen and couple that with eating the material of lysed cyanobacteria, so that when you finally get to the layer of purple sulfur and green sulfur bacteria, these anoxygenic phototrophs, this zone is much more depleted in oxygen because you had those heterotrophs kind of take it all up already in their respiration process and then release CO2. Therefore, the anoxic photosynthesizers are down far enough to get away from the oxygen and to access the sulfur produced by what's in our next layer, which is going to be sulfate reducers. We talked about these in a lot of videos of mine. Basically, these guys reduce sulfate to produce a whole bunch of sulfur intermediate compounds like sulfide, elemental sulfur, and other sulfur intermediates that the green and purple sulfur bacteria can then oxidize back to sulfate for the sulfate reducers below them once more. This is sulfur cycling. This cycling occurs in this anoxygenic phototroph zone as well as sulfate reducer zone, and this transition between the two. And if you're wondering, okay, so they have access to the sulfur and they kind of get away from the oxygen because they're deep enough, but since they're so deep, does sunlight even reach that far since they are still photosynthesizers and they use photo or light? Well, yes, it actually does. And what's cool about these guys and about biological evolution as a whole is that cyanobacteria in their oxygenic photosynthesis use certain wavelengths of light, whereas anoxygenic photosynthesizers like purple and green sulfur bacteria use totally different wavelengths of light. And therefore, the light that does penetrate down to where those guys are is actually the type of light they like. And so it works in their favor to be deeper down where the sulfur is, where the light they like is, and where the oxygen is not, and so on. And the bottommost layer or few layers in the cyanobacterial mats is anaerobic heterotrophs. These are the guys that they are heterotrophs instead of being phototrophs and using light to make energy. They are actually just eating up all the organic material that the anoxygenic phototrophs create when they die and go down to the bottom layers of the mat. And so they eat those guys and they couple that with a couple different metabolic pathways. One is sulfate reduction. So SRB are sulfate reducing bacteria shown here, and they couple their organic matter uptake with the reduction of sulfate produced by these anoxygenic photosynthesizers by sulfur oxidation. And they reduce the sulfate back to the reduced sulfur and sulfur intermediates that those overlying guys eat again. So it's a cycle again. And with this, they also produce carbon dioxide with the organic carbon that they originally took up. The methanogens, on the other hand, take up the organic matter as well as the carbon dioxide and produce methane. And these are completely anaerobic heterotrophs. They do not like oxygen and therefore naturally zone to the bottom layers of the mat, which, as it turns out, works perfectly for all of the other mutually benefiting communities of bacteria in the mat. And that is cyanobacterial mats for you. But photosynthetic mats don't only include those that contain oxygenic photosynthesizers like cyanobacteria, but they also include mats with only anoxygenic photosynthesizers like the purple and green sulfur bacteria we talked about on the previous slide. So imagine the same mat we just talked about, but without the top two layers of cyanobacteria and aerobic heterotrophs. Where would these types of mats form? Well, if you think about it, they include all the other types of bacteria except those that need oxygen, and therefore would probably form in somewhere that had sunlight for the photosynthesizers, but no oxygen. However, like you can imagine, somewhere where there's sunlight but no oxygen is not super common on modern Earth. But on early Earth, this would have been the norm prior to around 2.4 billion years ago. What happened around that time? The great oxidation event. I have a video about that if you want to check it out. I'll link it up here. But that is when oxygenic photosynthesizers evolved and began to produce oxygen, building up oxygen in Earth's atmosphere and oceans. And this eventually led to the evolution of eukaryotes and macrofauna. But prior to 2.4 billion years ago, sunlit environments with no oxygen would have been great for these types of anoxygenic photosynthesizers. And so it's possible that these could have been the first microbial mats ever to form on Earth. But it's still uncertain whether oxygenic photosynthesizers versus anoxygenic photosynthesizers evolved first, etc. But I'll talk about all those possibilities in a future video when I talk about the evolution of photosynthesis. Moving now to chemotrophic mats. These are those mats that would form in the absence of sunlight, where a lack of macrofauna would mean great for microbial mat growth because the macrofauna wouldn't munch down all of the microbial mats that begin to grow. But because there's lack of sunlight, that would mean no photosynthesis, which would mean no oxygen. However, like I talked about previously, for anaerobes, this is great news. They hate molecular oxygen. And so a few different types of bacteria would thrive in these types of mats. These include colorless sulfur bacteria, which are sulfur oxidizing bacteria that aren't phototrophs, so they don't have to have sunlight, but they still oxidize the sulfur that is produced by SRB, sulfate reducing bacteria, in these anaerobic environments. And underlying colorless sulfur bacteria layers in these types of mats would be methanotrophs and SRB. Once again, the same type of bottommost layers that we saw in cyanobacterial and other types of mats would also be present here because, again, these guys don't need sunlight and don't like oxygen, making them perfect for these types of chemotrophic mats. And these types of anoxic environment chemotrophic mats actually dwarf their oxic counterparts. And why would this be? Well, it's because of the lack of macrofauna. And now that we've talked about mat development and the types of microbial mats that can form, let's talk a little bit about how mats can be preserved as microbialites, lithified microbial mats, in the rock record. This lithification typically occurs with the help of minerals. And so it turns out to be a mineral organic matter layered structure, kind of like shown here to the bottom left, that is either lithified by silica, calcium carbonate minerals, or even sometimes pyrite in a process called pyritization. I talked about that fossilization technique in the 5th biomineralization video if you want to check it out. But these are the common mineral types for lithification of microbialites. And there are a couple different microbialite depositional environments that we'll talk about. The first one being thermal spring environments, in which microbialite deposits form under high temperatures. Therefore, this checks the box of being prohibitive to macrofauna and allowing microbial mat growth. And also, because it's higher temperatures, aragonite is the primary calcium carbonate mineral that would be present there, as well as amorphous silica could also be present and contribute to the lithification of microbialites in such environments. Which one ultimately wins the mineralization battle depends on the relative saturation states of aragonite and amorphous silica in the spring environment. And these mats would contain somewhat thermal or maybe mesothermophilic bacteria and archaea because the temperatures are high enough to be a little bit prohibitive to the more normal temperature liking bacteria. And so the bacteria have to be a little bit thermophilic to actually thrive in these types of environments and grow microbial mats. And the textures of the mat would just be basically dependent on what types of microbial species were dominant in the mat, because textures of microbial mats can really vary, as we've seen in these pictures. The second major type of microbialite depositional environment is the inter and subtidal environment, which promote stromatolite deposition, in which cyanobacteria dominate, trapping detritus in their EPS to form cohesive mats. And these types of mats are typically calcified, meaning they're held together by calcareous or calcium carbonate cement. Typically, in these lower temperature environments, calcite would be the major calcium carbonate mineral to lithify these microbialites, as opposed to the aragonite we saw in the high temperature environments that lithified the microbialites in the thermal springs. And it isn't actually the cyanobacteria that promote the calcification of microbial mats in these inter and subtidal environments. Actually, cyanobacteria, yes, they do trap detritus in their EPS, but since their metabolic pathway promotes a decrease in pH, they actually prohibit calcite deposition, whereas the underlying anaerobic heterotrophs facilitate calcareous cement formation because their metabolic pathways promote an increase in pH, which is favorable for calcite deposition. Once the calcareous cement begins to form, it lithifies the detrital sand grains that had been trapped by the cyanobacterial EPS and the organic matter together and forms a sandy calcareous microbialite. The last thing I just want to briefly go over is that it's important to note that microbialites such as stromatolites are different than thrombolites. You may have seen this in a book or heard this term before, thrombolites, and that is because these are similar to microbial mats and look a little bit like them structurally, but as you can see, they can be distinguished from microbialites by their massive size and their macroscopic clotted fabric. So you can see how it's not just a layered mat here. It's got this clotty texture. It's all jumbled up, and that is because macroorganisms disrupt the framework of microbial layers, and these thrombolites stromatolites are disrupted by macrofauna. That distinguishes them from stromatolites and other microbial mat formations that are purely or dominantly microbial rather than microbial? No, not macrobial, macroorganism dominated. And like you'd probably imagine, thrombolites are more common in environments inhabited by macrofauna and therefore are more common today on modern Earth, whereas on early Earth, stromatolites and microbialites would have dominated prior to the evolution and biodiversification of macroorganisms. But I think we can all agree that all types of microbial or even macrofauna disrupted mats are just so cool. But in terms of the upcoming videos, we have, like I talked about, Microbial Zonation Part 2, which is going to talk about marine sediments, specifically organic sedimentation and diagenesis. I've had a few people requesting diagenesis here before, so this is where we're going to talk about that. And then the following one will be the Part 3 Microbial Zonation video over, again, marine sediments, but in this case, oxic, suboxic, and anoxic sediments, and the difference between those types of environments and the sediments that are deposited there, as well as the microbes that live there and how they affect sedimentation. And lastly, for this video, my major reference in making this video was Introduction to Geomicrobiology by Kurt Konhauser. This book is linked in my description down below. Oh, I just noticed it has stromatolites on the cover. So there you go. It's really quite a perfect fit for this video, if you will, and this is linked down below, as well as other supporting references I'm using to make this and other videos if you want to check those out as well. And last, before I let you guys go, I'd love it if you would comment down below for me any geobiology topics you'd like me to go over in the future, or any other type of topic you'd like me to cover on my channel in the future. Just comment that down below so that I know what you guys will be interested in future videos, and that would be great. I can't wait to see y'all's comments, and thanks again for watching. I'll see you guys next time. Bye!